The title of our study is Ecophysiological and Genomic Characterization of Comox strain Nitrospira species BO4. In our study, we enriched Nitrospira BO4 from freshwater Lake Baroque, and the enrichment contains a monoclonal Camomox nitrospira together with several heterotrophic bacteria. In our study, we characterized the growth of the Camomox enrichment under different conditions, namely ammonium concentrations, pH, light, and nitrite as substrate, as well as um, analyzed the metagenomic sequence of the enrichment to obtain metagenome assembled genome, and also analyzed the metagenome assembled genome of the Comomox nitrospira. We firstly performed growth experiments of the Comomox enrichment under different ammonium concentrations ranging from 25 micromolar to 3 millimolar, and we observed that their growth rates were pretty comparable to the growth rates of the freshwater ammonia oxidizing archaea AC2. As compared to the AOA and Comomox, the growth rates of the freshwater on AOB enrichment G57 increased with increase in concentration of ammonium. We then performed growth experiments at different pH ranging from 6 to 8.5 with BO4 Comomox, and observed that their growth rates were again comparable to the growth rates of ammonia oxidizing archaea AC2, while the growth rates were much lower than the growth rates of AOB enrichment G57. We then incubated the enrichment in white light, dark, red, and blue light to observe if they have an impact on their growth. And it was pretty interesting that to observe that the Kamamox enrichment was significantly inhibited. Their growth rate was significantly reduced in white light incubation, as compared to dark incubation. We observed that the red light did not have any effect on the growth rate of the Comomox enrichment. Um, and in blue light incubation, the growth rate was completely gone. The Comomox enrichment was not able to grow at all in blue light incubation. We then incubated the Comomox enrichment in nitrite only at a concentration of 0.1 millimolar nitrite, as well as in a combined substrate incubation of 0.5 millimolar ammonium and zero point 0.1 millimolar nitrite to observe if they were able to oxidize nitrite as substrate. We did not observe any oxidation of nitrite um, in both conditions, while the enrichment completely oxidized ammonium to nitrate. Um, Then we performed the metagenomic sequencing of the BO4 Comomox enrichment and analyzed the data on K-base. We were able to obtain a metagenome assembled genome of the Comomox nitrospira, which we have named nitrospira species BO4 here. And the genome was closest related to Candidatus nitrospira nitrificans and Candidatus nitrospira crafti. The genome In the genome were detected all the genes required for ammonia oxidation as well as for nitrite oxidation. However, we were not able to detect any nitrite or nitrate transporter in the genome, and which aligns pretty well with the inability of the Comomox enrichment to oxidize any nitrite in the growth experiments. We then constructed a pangenome of 9 nitrospira, cultivated nitrospira, of which 5 red ones are the Comomox, while the blue uh, are the canonical nitrospira, nitrite oxidizing nitrospira, and the pangenome here on the top left shows all the genes that are present in all the genomes, um, and all the portions that are lightly colored are the parts where that represent the genes that are not present in the genome. Um, and the heat map on the right shows the average nucleotide identity of the genome, and the phylogenetic tree on top of the heat map shows the clustering of the genomes. And it is pretty cool to observe that all the cultivated common are clustering together, while all the canonical nitrite oxidizing nitrospira are clustering together, and our BO4 enrichment is also clustered together with the four other cultivated Kamomox nitrospira. So to sum up, we were able to figure out that our Kamomox enrichment has a growth rate similar to a freshwater mm, ammonia oxidizing archaea AC2, while it's different from the growth rates of the freshwater ammonia oxidizing bacteria G57. The enrichment is inhibited by white and blue light, and it's not able to oxidize nitrite, externally supplied nitrite, and possibly in lack of nitrite transporter. And the genome aligns clusters closely with other cultivated Kamamox nitrospira.